So welcome everyone. My name is Sofia Protzinhos, and together here with my colleague Rolando Garcia-Milian from the Cushing Whitney Medical Library at Yale, as well as Molly Knapp, Rana Morris, and Kate Majewski at the National Library of Medicine, we welcome you to the series of talks dedicated to explore the evolution of bioinformatics services in libraries. The idea to organize this series arose from the celebration of the 10th anniversary of the Bioinformatics Support Hub here at the Medical Library, which led us to ponder over the collective past, present, and future of bioinformatics services in libraries. And to share just a little bit of our story, the Bioinformatics Support Hub was launched in 2014 by my colleague, Rolando Garcia-Milian, focusing on the retrieval of molecular information from knowledge databases. Between 2015 and 2017, the service bolstered its collection development, specifically through the licensing of software designed to enhance data processing and analytic support. The years of 2018 and 21 marked a period of scaling characterized by the introduction of a peer-to-peer teaching program, the establishment of the Symbonus Fellowship, and the creation of a new permanent position. That is when I was hired, dedicated to education of command line tools. And from 22 to 24, our efforts have been directed towards establishing and strengthening collaborations with various institutional departments and course facilities, further solidifying the service integral role within the academic and research community. So it was based on this reflection process that we decided to join forces with the National Library of Medicine and organize this series of 7 roundtables, basically, around bioinformatics services in libraries. And we are very excited for today's sessions dedicated to learn how bioinformatics librarianship can be done beyond the academic environment. And to help us with that, we have invited, we have 2 speakers, mostly 3 speakers, but 2 of them will present together. Julianna Pakstis and Nicole Feldman from the Children's Hospital of Philadelphia, and Dr. Joubert from the National Institute of Health Library. And I will introduce Julianna first, and then Doc. They will all have 20 minutes to present each, and so I will introduce Julianna first and Nicole Feldman, who are both working as part of the Arcus initiative, which aims to facilitate the management of research data, including omics data, during every stage of its life cycle. Julianna's work focuses on describing archived research data from across the Children's Hospital of Philadelphia. She designs and produces metadata records and schemas for collections in the Arcus archive. And prior to this role, Julianna was also cataloged audio, visual, archival, and rare book collections at Temple University, the University of Pennsylvania, and the University of Chicago libraries. So Nicole's role focuses on archiving and preserving high-value research data sets from across the Children's Hospital of Philadelphia, and her role requires capturing in context emerging data types and standards in the bioinformatics and clinical big data domains. Prior to joining CHOP, Nicole worked as a digital asset manager in media publishing or highlights for Children's and Condé Nast. So with no further ado, I will give the stage to Nicole and Julianna. I will stop sharing. And you guys can take the virtual stage. Thank you so much for accepting to present today.
Yeah, thank you for that introduction. Everyone should see our title slide. Is that what you're seeing? Okay, I see some shaking heads. All right, thank you for that introduction. Thank you everyone for joining us today. You're going to hear some repeated information, but we are going to just go through what Nicole and I here do at the CHOP Arcus program, talking through how our bioinformatics work and support has evolved as part of that program. So I'm Julie Pakstis. I am a metadata librarian. I'm joined by Nicole Feldman, a digital archivist. We have the overview and objective for the session. We are going to talk really about our experiences and give specific examples of bioinformatics librarianship and support that we provide through archiving for biomedical research data. Like I said, we're going to talk fairly extensively about Arcus, what it is, what we do, and the library science team of which Nicole and I are a part in depth. We'll briefly mention some parallel and inspiratory efforts for Arcus and for the library science and archives component of it. And then the majority of our presentation is going to be the bioinformatics specific components of Arcus. We'll go through our data catalog, a process for biosample matching, our new initiative for AI and ML model cards, and then some of our omics tools and research data management services. So first up, I'm going to talk through an intro to Arcus. This is a somewhat unique program. We are situated within the Research Institute at the Children's Hospital of Philadelphia called CHOP. We are a multi-year strategic initiative that contains a suite of tools and services with the aim of supporting research efforts however we can for researchers, but primarily through allowing them to explore available data, see overlaps among data sets, clinical data sets and research data sets, allowing them to build new cohorts, and to determine where data or physical samples or additional resources are available for new research projects. So within that broader mission, the Arcus Archives and the library science team collects, organizes, stores, and describes data and tools, so contextual information, from all across the CHOP Research Institute. So we do have a big focus on omics data, and we will talk more about that, but we also have clinical and tabular data sets collected and used in the context of research, imaging data, survey data, any kind of data that comes from a research project in the Research Institute falls into our collecting strategy. So we will be able to share out these slides, and we have a link here to some resources about Arcus and some tools and services that we provide internally at CHOP, as Arcus is primarily a CHOP researcher service, and that is for the sensitivity and privacy of the biomedical data that we have. However, we do have some limited publicly accessible resources. We're starting publicly accessible collections through collaboration with Harvard Dataverse as well. And we do allow collaborations and non-traditional personnel onboarding for our CHOP collaborators. So that is something a little bit different from a traditional library environment where there's a little bit more restriction, but you will be able to see the resources at this link. So when we're thinking more about like Arcus is an initiative, it is a suite of tools. What does it actually do and who is involved in it? We're kind of using the research life cycle as described here to talk through some of the specific services and expertise that we provide. So we do have a library science team with seasoned digital archivists, librarians. Then we also have an education team with educators on things like SQL and Python, for example. We have privacy analysts and data analysts. We also have dedicated programming staff to help build all of the various tools that you'll hear about today. And what our overall aim is to support researchers from the discovery of available data for their project through to the completion, publication, archiving, and sharing of that data. So we kind of start with something like data management and sharing planning services. We offer tools to help explore CHOP data, again, clinical data, all kinds of research data. We offer secure computational lab environments for both synchronous and asynchronous research collaboration. So that's kind of in the analyze slice of this pie here. And then as we're moving up to archiving and sharing, of course, we have the Arcus archives where we do data archiving and provide archiving services. And all throughout this life cycle, through kind of an initial delivery and possible additional deliveries, researchers have access to CHOP patient data, to archived research data, to clinical data cohorts, GIS data, NLP processing data, omics data, actigraphy data. So we're really trying to get as much and as varied types of data in front of researchers who are permissioned to see it as possible. And then we have our education services that also work kind of alongside library and alongside the team to help even new researchers who maybe have never used a SQL query before to explore their clinical data through to learning new skills in how to analyze a data type that maybe they haven't worked with before. Also wanted to shout them out here because they are responsible for this lovely graphic. So Arcus is quite a broad program, but Nicole and I are part of the library sciences team, which here has 5 people. Anae Bella is our supervisor, and then we have 2 digital archivists, Nicole Feldman presenting here today, as well as Allison Olson. We have myself, who's a metadata librarian, and then Jenny Lukey, who is our data catalog and discovery librarian. And we are the team that is responsible for the process of archiving research data. So we also publish metadata about that data to our gene data catalog, which is one of the bioinformatics components that we'll talk about. We facilitate redelivery of that data into the Arcus computational labs for further research. And then we provide research data management support as well. So we essentially follow, again, within the overall research lifecycle, this cycle here from research data creation and collaboration through to reuse. And we do that by partnering with the study teams and the bioinformaticians who work on their own research projects and protocols. So we're assisting them in research that they're spearheading, essentially. The digital archivists work directly with those study team members to understand what data is in a collection, what data might need to be part of a collection, what the file types are, various content and contextual files, any technical requirements or workflows that would make reuse possible. So we're documenting the entire research effort. And from our first point of contact with the teams, we do talk about appropriate organization of research data so that it is more cleanly transferred into the archives. So that's our archival process here. Once the information is archived, then it goes over to me, who's the metadata librarian, and I apply metadata to that archived research data. And this is pretty extensive metadata, ranging from a really generalized summary of the research project itself down to information on individual files in the collection and accounting of what participants or patients are represented by individual files in the collection. So once that metadata is complete and it is approved by the studies team, which is another area of collaboration with them, then we push just that metadata out to the gene catalog. Once there's metadata about a collection in the gene catalog, other researchers or even the originating researchers for a different project are able to discover that data and request it for reuse. So that will then bring us kind of back to the top of the diagram where there's additional research data creation and collaboration, and our research data management services help researchers who've been through the process understand the data that they're getting and put their data in good shape for completing this next project and re-entering the cycle. So we want to do that, obviously, to provide data back into the archives and to really position ourselves as the central repository for data at CHOP, but also, of course, to help our researchers meet external funding requirements like the DMSPs for NIH grants, for example. Here I just wanted to mention, as we've kind of talked about the context for Arcus and our team, just some of the parallel and inspiratory efforts from when Arcus was started. So Arcus began in about 2016, 2017, and definitely has evolved since then. But since the beginning, we knew that we wanted to be FAIR compliant, and we also wanted to be able to leverage and interoperate within existing systems for biomedical data and other standards that had already existed. So around 2017, 2018, we did kind of a survey of what was out there, what we could utilize or adapt on, and I just pulled some examples like the database of genotype and phenotypes, dbGaP, data for the common good, data site and data tag suite that really position data as kind of the primary objects for sharing, for attribution, and for reuse. So we wanted to call those out here. In the next bit of slides, we're going to go on to the bioinformatics components of Arcus. So each slide or set of slides is going to represent a slightly different example of the kinds of work that we do in Arcus. So first up, I'm going to talk more about our gene data catalog that I mentioned in the overview of the library science team. So this was officially launched in June 2023, so a little bit over a year old. And it is an enterprise data catalog. It's called Gene. This catalog is built on the Elation platform. Gene is our CHOP branding for it. And Arcus works alongside a collaborative group called the Gene Custodians with members of other teams in the Research Institute. So namely the Data and Analytics team, or the DNA team, which is within the Digital and Technology Services group. So what this means, this collaboration enables us to have a really, really broad collecting policy and a really broad set of data represented in Gene. So kind of on the DNA side, we have databases and data warehouses where you can see what tables exist in those services with a description of which columns are associated with each table. We have analytics assets that allow end users to explore, filter, analyze, and visualize data. Then we also have like a very wide swath of documentation, things for best practices and standards that are available about all of the data and analytics assets from that side of the Research Institute. The other big piece of the Gene Data Catalog is the representation of archived research data in the Arcus archives. So all of the metadata that I create from all of the collections that the digital archivists bring in to the Arcus archives is represented in Gene. We generally have two types of collections that come into our archives. One is a research collection or a data set, which this is the broadest type. This is generally like the results of a research project, including the data itself, any code objects, data dictionaries, protocol, consent, lab documentation, notebooks, all of that type of stuff. So that can be like omics, imaging, if there are REDCap surveys. All of those kind of things can go into a research collection or data set. We also have reference cohorts, which is a definition of a clinical cohort that can be used in the research setting that identifies a specific group of participants or patients who match that definition. So some of these have really complex inclusion-exclusion criteria where it's useful for a researcher to be able to understand what the query pulls and utilize that without needing to rewrite the query from scratch. So I was careful to mention that the Arcus archives in Gene is represented only through metadata. So we don't show the actual data even to CHOP researchers. It really is a metadata representation of what can be requested in the archives. And of course, this is for privacy considerations, for making sure that all of the regulatory and IRB considerations are checked off before a researcher has access to data. So because of that, we do have an archived data delivery pipeline that takes the request, reviews the request, sets up the computational lab. Again, this is a little bit simplified version of the pipeline, and then delivers the specific data that the researcher needs and that overlaps with their cohort into their computational lab in Arcus. So Gina is how we view the metadata, and then we have a request process that gets the data in the hands of the researchers. So a completely different example now is our biosample matching pipeline and services. So this image depicts the overall biobanking landscape at CHOP and also kind of notes how Arcus fits into it. So if we're looking at the lower right-hand corner here, we see Arcus, and then we see this orange box for TRIG, which is the Translational Informatics Group within the same department as Arcus, the Department of Biomedical and Health Informatics. We partner together to represent overlaps in clinical and research data and to find participants who match that definition, who also have biosamples that are stored in the CHOP Biorepository Resource Center, or BioRC. So TRIG does ad hoc data integration, and because of that, they work also like Arcus with individual research projects and protocols, and they have an operational administrative database that can query across all of those projects to find biosample or sample information and availability. Arcus we have, of course, our data repository and archives, but we also do honest brokering for research projects, and we have something called Arcus Cohort Discovery, our ACD, which is a tool for inputting inclusion-exclusion criteria for a cohort and getting counts matching patients fitting that criteria back. So when Arcus and TRIG come together, that infrastructure allows us to utilize TRIG's operational biobank to get information for up-to-date sample metadata for cohort discovery to see the overlap between a clinical definition of a cohort and who has biosamples. The researcher will first get the counts of the biosamples, and then we have a process where Arcus as an honest broker can work with TRIG to identify the participants who have that biosample, and then to match the requesting PI to the PI who deposited the sample in the Biorepository Resource Center storage. So when all of this comes together, what it looks like is a connection between two PIs, of course, one looking for physical samples, one who has the permission to give and to furnish those physical samples. And of course, oftentimes we do want to make that connection, but what's really critical here too, and where Arcus kind of comes in a little bit upstream, is that if we're working with a PI who is looking for a physical sample, like a blood sample, for example, because they want to do whole genome sequencing on that blood sample, and Arcus knows as an honest broker who that participant is, and they have all the correct permissions, and in the Arcus archives, if we have the whole genome sequence for that participant or group of participants, we can say to the researcher, we can actually just give you the whole genome sequence file rather than you needing to spend the resources and use the physical blood sample to rerun and get the whole genome sequencing that we already have. So it's kind of a cost-saving, time-saving collaborative measure. And then, again, switching gears one more time here, we are, like a lot of other institutions, looking at ways to integrate artificial intelligence and machine learning into our workflows. One way that we're doing that, specifically with metadata, is through standardizing model cards. So we want to have one document and one standard in Markdown or in a doc format that describes a model that is being used or created at the Research Institute at CHOP. So the chart here kind of shows the proposed details of the types of fields that we want to collect. But if you're working in this space or doing something similar, you may know that there's not really a full industry standard for what should be collected within a model card. So the way that we pulled this together was looking at the things like Google, things like MetaLlama, and a bunch of examples on Hugging Face to figure out where the overlap exists and to figure out what needed to be augmented for the biomedical space. So you can see something like model protected health information, or PHI, is not often found in industry models, not working with biomedical data, but it's definitely something that is very important for us to reference and to know about. This next slide here talks about those same fields, gives a little bit more instructions to the researchers who would be filling out this model card, and provides additional examples of the types of things that we will be collecting. So this is definitely ongoing work, and something that we're exploring in parallel with creating like an annotation card for annotated data that feeds into model or machine learning research. So we're going to switch gears again, and I'm going to actually pass over to Nicole to talk about some of our omics tools and then our research data management services.
Great. Thanks, Julie, for presenting so far. So I'm going to talk by speaking about our omics data services. Thanks. So clinical and research teams across CHOP have generated a multitude of omics data from patient samples. However, it's historically been difficult to identify existing omics data and request access to it. Arcus aims to archive and harmonize these data and make it easily available to all researchers at CHOP, in concert with the vision of Arcus that Julie laid out so well in her presentation, to make heterogeneous research data broadly available and reusable for researchers. So we've primarily collected whole exome sequencing and whole genome sequencing so far, but we're also starting to collect small subsets and are further expanding into other data types like transcriptomics and RNA-seq, metabolomics, and proteomics. Julie mentioned how we have a lot of different teams supporting Arcus, so we work really closely with our applied data science omics teams, and they assist in multiple processes, such as the data contribution and archiving, the development and harmonization of bioinformatics workflows, and assist with requesting access to omics data in Arcus, as well as its interpretation. So a tool we've worked really closely with our omics team on is the Rare Variant Browser, which is a collaborative project between our group at CHOP and the Center for Applied Genomics, which is a large core sequencing facility at the hospital. The CHOP Rare Variant Browser serves as a proof of concept to explore the feasibility and power of combining CHOP patient genomic data and making it visible to our entire community. The browser allows the community to query data from a large group of CHOP patients to identify variants and genes of interest, paving the way for future research discovery. So the exome and genome sequencing I mentioned in our previous slides that comprises the Arcus archives was aggregated, normalized, and annotated to provide a readily accessible variant browser websites. The variants in the browser are derived from close to 20,000 exomes, a little over 2,000 genomes, which are about half from our group and half from CAG. And I would say the data we collect in the Arcus archives, it's a mix of some of those core sequencing and service providers on campus, as well as several longitudinal research studies and biospecimens that are more disease focused and continually enroll patients and track them throughout their disease trajectory. Variants in the browser are based and have been aligned to the HG38 reference genome, but a lot of the sequencing data we collect is older, especially for some of these longstanding research studies, and aligned to HG19. So we work with our applied data science team on realigning everything to the HG38 genome. And only variants with a nomad allele frequency of less than 1 are listed, and all variants are annotated with VEP, or the variant effect predictor. And users who browse the tool can then request access to a scientific lab with data discovered in their selected filters, sort of like a similar service to the BioRC sample matching that Julie mentioned earlier in our presentation. So this next slide is a good visual depiction of sort of what I explained the variant browser tool is. So you can see in the data sources, I mentioned the Center for Applied Genomics, who is our partner in this project, but then DGD, they're the Division of Genomic Diagnostics, so they're one of the largest clinical sequencing providers on campus. And then Engine is a large epilepsy repository, and BDB is a birth defects biorepository, which is primarily patients born with cardio birth defects. And you can see in the browser, we currently have over 42 million variants. Currently, as I said, the browser is sort of in still a concept, like beta proof of concept phase. But our team is now transitioning from sort of these manually composed GTK best practices workflows to Illumina's Dragon software that will allow for much better scalability and automation and workflow transparency and reducibility as we capture more omics data and hopefully have more users using the tool as well. So the last, yes.
Oh, I'm sorry. I just wanted to tell you, you have about 2 minutes.
Oh, okay.
Thank you.
So the last area we focus on that I know a lot of other presenters have also mentioned in previous presentations is research data management. So our resources are linked there as well. So we provide guidelines and project documentation, file naming, data dictionary, process documentation, version control, file organization according to the project template, which is a file directory structure. We also collect all our research data sets in anthologies, and also how to use REDCap, which is the preferred platform for like survey collected research and other database derived research at CHOP. So we also use Jira Service Desk for project management and customer support at Arcus. So that also allows us to connect with users on research data management topics and support issues. As many other presenters have noted too, we also do a lot of DMSP and publication support. We've conducted about 60 consultations since the plan went live in January 2023. We have linked here the DMP tool, which is a great support user through California Digital Library that we set researchers up with. We also provide guidelines on things on grant language for those using Arcus during pre-research or as a platform for research, and citation formatting and use cases, since there's so many services that Arcus supports. And lastly, just wanted to focus on some of our outreach and teachings that kind of continue to evolve as we connect with more groups at the Research Institute as Arcus becomes a more established program. So Julie mentioned we have a data education team who more tries to upskill our workforce in Python, R, and SQL and promote data science fluency. But we sort of worked with them on developing interactive LeaScript modules, which is a form of Markdown that allows you to publish instructional content on that file directory structure I mentioned before. We just started teaching a quarterly course through our Data Trust and Literacy Office on the organization and management for files, data, and metadata. That's allowed us to connect with a lot of clinical research coordinators and other research support staff at CHOP. And our Office of Academic Training and Outreach, they hold several responsible conduct for research roundtable discussions every year. So our team has been facilitators for their panel on NIH data management and sharing plans, and that's more allowed us to connect with research trainees, postdocs, fellows, people in their K award or T32 period. So we're just continuing connecting to all different kind of user communities within the Research Institute and hospital. And I would say we had our first responsible conduct of research round table of the year a few weeks ago, and we noticed year over year that really the familiarity and enthusiasm for the data management sharing plans had really increased. So we were really encouraged to see that. And that's the end of our slides. I'm not sure if we're doing questions at the end.
Yeah, we will do questions at the end.
Oh, okay, great.
Yeah, thank you so much, Juliana and Nicole. And now I will briefly introduce Doug Joubert from the National Institutes of Health Library, and he's the chief of the Research Analytics Branch, where he oversees the strategic alignment of the NIH library services in bibliometrics, bioinformatics, biostatistics, and data visualization to support the mission of the NIH. Thank you so much, Doug, for joining us.
Sure, it's great to see you again. Can you guys hear me okay?
Yeah.
Awesome. And looks
And
like
we
my
see your
slides. Okay, thank you so much. So I'm going to talk a little bit about what we do here at the NIH, a little bit about our users, about our service model, and staffing and resources. So in contrast to all the wonderful speakers that have gone before me, I'm approaching this as sort of a person who leads a team, not necessarily a practitioner anymore. So I will talk about staffing and resources and some difficulties with recruiting and retaining staff. As others have noted, nothing happens in isolation. So I'll close with our NIH partnerships and collaborations. There is a PDF version. Let me just go ahead and just drop my profile information in here. If you want a PDF version of the slides, just reach out to me because there's a lot more stuff in there, some goodies in there about data training and our bioinformatics training. So if you've never been to our beautiful Bethesda campus, this is a wonderful aerial view of this. I do always like to start my presentations by acknowledging that this is the ancestral territory of the Piscataway Indian Nation and Tayac territory. And I think it's always important for us to remember the original stewards of this land. So having said that, we are focused primarily on the intramural research program. So you can look at the numbers here. I'm not going to read these out to you in terms of our client base, our customer base, because some folks on this call probably are, we're all in library land, right? So I do think it's important to also say that we have 20 interagency agreements with the operational divisions. So those we're part of NIH, which is part of HHS. So we have interagency agreements with a number of different sort of HHS or outside of NIH operational divisions, and we provide resources and staffing to them as well. And at least in terms of where we sit, we're in the Office of Research Services. So this is my team. Some of these are typical to most modern libraries like data services, bibliometrics. We are unique, and I consider myself very, very blessed in terms of who I'm able to hire. We have full-time FTE contractor person, Pam Burrows, who provides support for biostatistics. And these lovely little AI-generated images are representing the 2 data science positions that I am currently hiring for. So I'll talk a little bit more about those later. So as the previous presenters alluded to, and many libraries practice this, we kind of we meet the researcher wherever they are in terms of the data lifecycle, from data generation, collection to analysis, interpretation, and sharing. Both in terms of our data services and our bioinformatics, we generally get people when they're downstream. So folks would come to us with the data, and they're looking for meaningful ways to either represent that through a visualization, or they want to analyze it using some of the biostatistical tools that we provide access to, including PAM. So the 2 primary drivers of our service model are just leveraging NIH library tools and expertise to support data analytics, data visualization, and bioinformatics. And because we're part of the NIH, we support the mission critical efforts of NIH and HHS. So in terms of bioinformatics, we support NIH researchers and clinicians who are seeking solutions for retrieving and analyzing biological data. Pam, our biostatistician, does provide some support for methodological considerations for research design, but both in terms of bioinformatics and data services, we generally are working with people after they have the data. And we do that through access to our software tools, access to our high-performance workstations, and our licensed resources. We do provide a little bit more coverage in terms of data because we have more people supporting our data services program. But in terms of our service model and what we provide is exactly the same for data services as it is for bioinformatics. So I'm going to get in my little TARDIS and travel back to 2009 and give you a little overview of sort of the genesis of our program. So our program was started in 2009 by Dr. Mehta Bhagwat. And Mehta taught at NCBI for many, many years. And if you've been doing this as long as I have, you probably remember in the late 2000s, NCBI was really at the forefront in providing education and training for people like myself that had somewhat of a science background and were interested in either supporting biomedical researchers or starting a bioinformatics program. And they called these the 4-pack. So it was primarily Mehta and Renata, if anybody remembers her. So when Mehta joined us here at the NIH Library, her service model was focused on really teaching. And initially, we focused on supporting sequence analysis. And then about a year after we started our program, we hired Lynn Young, who is from our mathematical statistical and computing lab here on campus. And she was hired to provide programming support for high throughput data analysis. Her focus, both from a practitioner and from a publication perspective, was on microarray and next generation sequencing data. And humbly, I say they were just such a great sort of power team. Mehta was the outward-facing face of our program. She was great in terms of user engagement. She loved teaching. And then Lynn was kind of behind the scenes. And although she taught and did many consultations, she was really doing most of the programming that supported the analysis that was being done here at NIH. So around 2010, we started we always had access to resources, but when I say we started adding licensed resource, we actually had a line item in our budget devoted to supporting bioinformatics. And I will say, as I go through these slides and I talk about what we had and what we have now, I am coming from definitely a place of privilege. We are well-funded as a library, and I do understand that not everybody has access to tools that we do. But when we started adding tools to our portfolio of services that we offer to our users, it was GeneGo, Metacore, Ingenuity, when it was still Ingenuity before it was part of QIAGEN. And it was really on connecting the published literature using pathway tools like GeneGo, Metacore, or Ingenuity pathway analysis. So kind of a trip down memory lane. Some of these products no longer exist. They will be recognizable to many on this call, I imagine. So we started with DNA and laser gene. I'm not going to read sort of the way these licenses operate because you're going to have access to the slides. Then probably around 8 months after we got DNA Star and laser gene, we got CLC Genomics when it was a standalone product and not part of QIAGEN. GeneSpring, Genomatic Genome Analyzer, HGMD, and that is the web version, still continues to be the web version. We don't have the version that you can download. Ingenuity Pathway Analysis, when it's a standalone product, and also the Varied Analysis Tool, which was part of IPA. Metacore and Genomic Analyst Tools, Partec Flow, and Partec Genomic Suite. So we started with Partec Genomic Suite because even though we started expanding the type of licenses that we had, so we had maybe licensing servers set up, folks initially in the beginning were really wanting to have one-on-ones with our bioinformatics support staff. So even though we had methods for accessing this stuff remotely, cloud wasn't around then, people still came to the library to use these tools. And it wasn't until 2012 that we started subscribing to Partec Flow. If you're not familiar with these tools, so Partec Genomic Suite is a standalone desktop client. Partec Flow allowed us for the first time to connect to our NIH APC high performance computing. And if I'm using any sort of terms that you don't understand, please put in the chat and I will try to define things because we have our own geek speak here at NIH. Then we did Pathway Studio, Protein Lounge, SeqMan, and then VeriSeq, which are part of DNA or LaserGene. In terms of the workstations that we set up, and again, these were kind of in the beginning, we had 2 Windows boxes and a Linux box. This might not seem like a lot of computing power now, but we purchased these, especially the Linux box, back when memory was much more expensive than it was. And as I said, people wanted to talk to Lynn and talk to Meta while they were doing their analysis, because especially in terms of Partech and QIAGEN, these were really new tools for folks. So we had 2 high capacity drives. People would check out the drives, take them back to their lab, load their data, and then bring that back to the library to do their analysis. So as you can see by the current snapshot of what we have on our workstations, and this is due to changing dynamics of the customers that we serve and also limitations in terms of funding, we have definitely scaled down in terms of the software that we provide. So now we only have 2 bioinformatics computers, Jump, which is the graphical view version of SAS, Partech Flow, Partech Genomics Suite, which is, I just heard from Partech is going away. They were just gobbled up by Illumina, so we're going to sort of stay tuned to see sort of what products Illumina is going to have around the corner. But and then Partech Pathway. On the Bioinformatics 2, we have CLC Genomics Workbench, which is now part of QIAGEN, the second version of Jump, and then the Bioinformatics Toolbox, which is part of MATLAB. And I also included, at least on my right of the slide, what we have installed in our data services workstation, because users are going to be users and we want them to use the tools that are most useful to them. Some people like to do things either in a Jupyter notebook. They like to use RStudio if they're using Bioconductor and Anaconda. So even though they're doing bioinformatics, sometimes folks just default for the data science workstation. So moving from sort of who we are, sort of the genesis of our program, and a little bit about the history of our resources, I'd like to switch topics and talk about recruiting challenges. These are not uncommon to anyone who's ever had to hire a bioinformatics person. In terms of the skills, I think it's in my experience, it's a little bit easier to find someone who has these wonderful sort of magical unicorn set of skills, right? The biological knowledge, the statistical expertise, and the programming skills. Back when I was sort of working more as a practitioner, there really weren't a lot of bioinformatics masters and PhD programs now. Now there are a lot more of them. So I think people are coming to work with all of the skills that we need. This is still, from my perspective as a hiring manager, definitely a high demand field. So it's very, very competitive. One unique aspect that's unique to us, and I apologize because this is not applicable probably to people in industry and academia. So the Office of Personnel Management, which is sort of guides our hiring, does not have what's called a bioinformatics series. So for the 2 positions, which I'm going to talk about in just a second, I had to be very crafty in terms of writing the job descriptions that are going to attract people that I want to hire, while at the same time not discouraging applicants who might not really see themselves working in a library environment. Obviously, we cannot compete against pharma when it comes to salaries. And location is a huge restraint for us in terms of staffing. The DMV, which is a district, Maryland, Virginia, we basically have folks that live like up to 100 miles around sort of the nexus of NIH, which is in Bethesda, all the way out. It's very, very expensive, and we no longer can pay for relocation expenses. I know I came from academia, well, that wasn't even on the table, but when you're hiring people from industry, they, sometimes there's the expectation that we're going to be able to relocate them. And this has become problematic if someone's just starting out of graduate school, or if they have a family, moving to a very expensive part of the country. It's just not an option for them. And I will say I've been doing this for 20 plus years, and I do see a gradual shift of newer PIs coming and sort of seeing themselves work in a library is not as difficult as it was when we started our program in 2009. So in terms of retention challenges, I'm not really going to read these because they're very similar to what I encounter in terms of recruiting. The two that I do want to focus on are the organizational constraints. As a supervisor, I humbly think that I do a good job of providing my scientific staff ample support to go out and just flourish and collaborate with other ICs or institutes and centers across the NIH, because I want them to still be connected to that larger scientific ecosystem. And most of my last 20 plus years have been in libraries, but I didn't start there. So I definitely can understand if you're coming from a science background and you are working in library land with library-based teams and the way libraries operate, that you can feel a little disconnected from the larger scientific community. So I think it's really important for supervisors to realize that and give our staff a lot of opportunities to collaborate across whatever campus you're on. So thinking of where we were in 2009, where our program started, to where we are in 2024. In 2009, very few institutes and centers had their own bioinformatics clusters or even had a computational biologist working with them. There was certainly no effort to coordinate trainings across the NIH. So we supported a lot of different researchers and a lot of different type of researchers. So back in my TARDIS to 2024, most of our ICs have some sort of staff devoted to bioinformatics, or they have either a cloud core or they have a data science team that's supporting computational biology. Through some work, I'm happy to say, that was done here at the library, training is coordinated across our ICs and the library. We're very fortunate to have Kathleen McLaughlin, our communications librarian, and she really helps us with cross-marketing and promotion with all the ICs. So we support a smaller set of researchers than we did before. And I don't mean smaller in terms of the amazing work that they do. It's just they don't have access to bioinformatics staff. So, and again, 2009, we supported a lot of researchers and a lot of different types of researchers. And generally in 2024, we support a smaller set of researchers. So I want to switch to the two data science positions that I'm currently recruiting for. Send me all your positive HR energy so I can get these folks on board in January. I will say as an aside, if you ever want to have a nice little laugh, go to AI and type in, give me a picture of a bioinformatics specialist, and just marvel at some of the images that you get. So the two people that I'm currently in the process of recruiting are brilliant scientists that have strong foundations in research and programming. I have a 3-year plan to move the library away from these very expensive licensed resources that we currently subscribe to. And I would really like, when these people are onboarded, to provide more support for Bioconductor, Galaxy, and open source tools. We definitely want to start supporting our customers for programming, machine learning, and AI. And then the last thing I want to talk about are our partnerships and collaborations. So as I mentioned, when we first started, there was not a lot of collaboration across campus. It's, our IT is still very centralized. The way that we hire people is still centralized. So there was a group called the Bioinformatics Coordinators Group that was started by a very ambitious PI. We're not officially sanctioned by anybody, but we're sort of this ragtag team that is really thinking meaningfully on how we provide both services, licensed resources. We talk a lot about contracts, how we sort of coordinate these efforts across campus. We also have a Bring Your Own Bioinformatics group. That is where I just had a PI reach out to me, a junior faculty member today reach out to me and didn't feel kind of comfortable bringing this up in her lab environment. She's new to doing pipelines, and she was doing some sequence analysis. So we try to help those people where we can, but in this sort of period of transition where I'm hiring these people, this Bring Your Own Bioinformatics is a group that I'm very comfortable sending junior researchers to. The same thing that we've done with the Bioinformatics Coordinators Group, we've also done with the Data Science Trainers Group. There's a wonderful person called Amy Stonelake, who is my counterpart at the National Cancer Institute. And what we do with the bioinformatics coordinator we also do with a data science trainer coordinators group. We try to partner with the Office of Data Science Strategy where we can. Our cloud computing environment is branded at NIH called Strides. When you get the PowerPoint, these blue colored text are going to be links out to the publicly available page where I was able to provide that. And I know we're kind of old-fashioned when it comes to listservs, but a lot of folks at NIH still use listservs, and I've just provided some public-facing websites so you can explore these groups on your own. A lot of very interesting discussions. So in addition to being a place for people in a very safe environment to ask questions like I'm trying to do the sequence analysis, I've really never done this before. Most of these groups also put on scientific talks here at the NIH as well. So again, PDF version of the presentation has more resources, also covers our data science, our peer-to-peer training, considering our wonderful host here. I had to sort of just highlight this article that was published. I know there are a lot of people that are doing peer-to-peer training, but the Yale Library is the first place that I've looked via this wonderful article. And then some resources you might want to explore from the Global Bioinformatics Education Summit, which is where I actually met Sophia in the spring. So that's all I wanted to talk about.
Thank you so much.
Sorry, zooming through that.
No, it was great. And unfortunately, we have like 3 minutes for questions, but yeah. So from our wonderful public, attendees, we have quite a nice turnout. So I open it for questions. Kate, do you see anything?
I do. Thank you so much to all of our speakers. We have a question from Elliot, a question for Doug. Could you talk a bit more about transitioning away from expensive licensed platforms and tools? Are you thinking about shifting to freely available community-supported ones?
Thank you for that question. I don't think we're going to get rid of everything, but I will be honest, our junior researchers, the postdocs, when we do, we're sort of drank the carpentries Kool-Aid here. So when we do our carpentries workshops, most of the tools that people are interested in are Bioconductor, Galaxy Bioinformatics. And the reason we started the peer-to-peer training is folks were coming to us and they were wanting support on specifically Galaxy Bioinformatics and Bioconductor, and unfortunately we did not have the capacity to do that. So I was Googling, how are other libraries doing training when you don't have the capacity? And that's kind of when I ran into what Yale was doing. And in the PDF version of the PowerPoint, I talk about some of the ways we've been able to sort of overcome that challenge. And it's developing partnerships across the NIH and having folks teach for us. I hope that answered your question.
Yeah, if I can add, Elliot, here at Yale, what we are also, this was not so much thought with that intention, but it's becoming a side effect, a positive side effect, which is when we collaborated with the course, that was our second session. One of the good points in collaboration was to learn about the pipelines that actually the core doesn't mind to make available for the researchers that use their service to sequence, then to use their pipeline to analyze the data. So, we have been, actually, Rolando has been very successful at guiding some researchers into using those pipelines. And so that kind of is giving us hope that we can make them, the researchers, more independent and using really accessible resources rather than Partech Flow, as wonderful as Partech Flow is.
All right. So time flies when you're having fun. our time is up. Thank you so much to Julie and Nicole and to Doug. I don't know about you, but I am planning, as soon as the recording goes up, I'm going to watch this all over again because there's so much great information, really, really useful to all of us. So thank you so much. Don't forget to fill out your evaluation. There's a link in chat and it will also pop up when you close the webinar. And there's also a certificate of attendance if anybody would like that you can download from the chat, which I'll also be sending out to all of our participants today.
And register for next session.
Thanks for watching. This video was produced by the network of the National Library of Medicine. Select the circular channel icon to subscribe to our channel, or select a video thumbnail to watch another video from the channel.